Hello to everybody. I hope that many of you who registered joined to this webinar. The topic of today's webinar will be about ticks and tick-borne pathogens, about understanding the risk and strategies for its prevention. And this webinar is connected with one of our active topic collections under MDPI. So later I will provide you more information in the chat about this topic that we have active, and you also can find more information about that topic. And if you have your own papers on this topic, you can submit it for publishing under our journals. Just short introduction about MDPI. MDPI is one of pioneers in open access publishing. MDPI was founded in 1996. Our headquarters are in Basel, in Switzerland, and our mission is to foster open scientific exchange in all forms across all disciplines. We have 472 diverse open access journals, and of those 463 are peer reviewed, and we have also 9 conference journals, and we are supported by more than almost 300,000 academic experts who share our mission, values, and commitment to providing high quality services for authors. I mentioned at the beginning that the title of this webinar is connected with one of our topic that is active, and the name of the topic is ticks and tick-borne pathogens. And at the moment we are running second edition of this topic. The first edition was very successful with 26 published papers. We hope that this second edition will be equally successful as the first one. The topic editors of this topic are Dr. Alina Rodriguez Melon and Dr. Alejandro Cabezas Cruz, who will be together with us here as the speakers. And Dr. Alejandro will be also the chair of the topic. And also here you can see the deadline for the submissions. And also you can see the link of this topic, but you will be able to Google this topic by the title of the topic, and you will find it at Google. Here is the program of this webinar. We will start with the chair introduction by Dr. Alejandro, and then we will have multiple speakers, starting with Mrs. Selma, and then we will continue with Mrs. Lianet. Then Dr. Alejandro will have his own part in these sessions. And after those 3 presentations, we will have a short period of time for the Q&A session, for your questions. And then we will move to the next session, starting with Mrs. Dagmara, then Mr. Kivo, then after them, Professor Marco Rogerio Andre. And then we will stop for a little again for a question session. And then we will have one final round of 3 presentations by Miss Ana Laura, then Mr. Julio, and Dr. Alina Rodriguez Melon. And after that, again, one short Q&A session. And then I will again give my word to Dr. Alejandro for the closing speech. The times presented here are in European Central Time, and you can see that the majority of the presentations will be about 10 minutes. You will find one icon that looks like this one, that is black Q&A, where you will be able to leave your questions. And please, when you are leaving the question, note the name of the person who are you targeting for the question, so we can provide you with a relevant answer. I mentioned that Dr. Alejandro Cabazes Cruz is our chair. He comes from University of Paris, yes. And now I will give my word to him.
Thank you, Alexander. This picture is like 20 years ago. I've changed a bit. Thank you for the introduction. Thank you, everybody. Good morning, good afternoon. So this is a really international event, like many time zones, people in many time zones being participating. We are very glad of that. And is it within the context of the DTP last year? That was a big success in Cuba. And then just you cover every area. I would just say that we are structured in 3 blocks. So the first block would be like a tick microbiome, then surveillance of pathogens, tick-borne pathogens, and then some control strategies at the end. I think mostly everything has been explained and detailed. So I would just give the floor to our first speaker, Salma Abdelani.
Hello, my name is Salma Abdelani, PhD student at Farhat Abbas University. Today I will take you into the complex microbial world of hyalomatics. This work, directed by Alejandro Cabezas Cruz, explore how ticks, microbiota, and pathogens interact, and how these interactions vary across seasons and between sex. First questions that arises here is why study ticks microbiota? Well, ticks carry a diverse and dynamic microbial community. It includes not just pathogens, but also symbiotes like transcellular. So, these microbes interact, sometimes helping and sometimes harming each other. And these relationships can shape a pathogen, survival, co-infections, patterns, and even transmission potential. So we focused on hyaloma excavatum ticks collected from Castle in Central Algeria. For the microbiome analysis, we sequenced 16 RNA from 21 female ticks. And for pathogen screening, we tested 6 in both males and females using high throughput PCR to detect 43 microorganisms using advanced techniques such as microfluidic PCR, developed by Sarah Moutsaï. Plus, it worked on sorry, what's happening here? Sorry, I lost stuff. So, I said plus network analysis, we were able to detect multiple pathogens. This gives us highly detailed view of pathogen diversity and co-infections patterns. For the microbiome, while overall thickness remains stable across seasons, the microbiome composition shifted considerably and show clear seasonal variation. So for sorry, I think I could not continue. I have problem in my breath. So can I just delay this seminar? I don't know. I could not complete like this. Hello.
Hello, hello. Yes, can you repeat?
Everything okay? Salma, would you like to conclude, or you have a problem with the presentation?
Yes, I have a problem with the presentation.
Okay.
I will try to complete.
Would you like to make some concluding remarks if the presentation just orally? Oh, it continues, right? So it's okay.
For the microbiome, while overall thickness remained stable, the microbiome composition shifted considerably. So, and show a clear seasonal variation with several bacterial roots displaying significantly different abundance between especially spring and autumn. In this networks, we see that Ferracicella was consistently present with particularly strong activity in summer. Rickettsia was.
Don't worry. So maybe you can just make the conclusion. So what's the main conclusion of what was the main conclusion of the study? And we can move forward in the section of answer and questions. We could explore a bit more that. Would you like to conclude what was the main conclusion?
Let's hear you.
Well, the main conclusion of the study of Salma was that there was a strong seasonal variation in the microbiome and also the frequency of tick-borne pathogens. And then there was most probably an association between the composition of the microbiome and the vector competence. So it's a dynamic process in which the microbiome of the vector is shaping at the same time, or at least that's the proposal, or what those results suggest, is shaping the presence of pathogens in the tick vectors and the capacity of those ticks to transmit the disease. So that's a key message in that work. So maybe we could, in the QA section, we can explore a bit more, and when we can pass to the presentation of Lianet. Thank you, Salma. Thanks a lot.
Hello to everyone. Today I would like to share with you how to use a network, how to use the network as a tool for the study of the microbiota and the pathogens interactions. We know that ticks are blood-feeding arthropods that transmit several pathogens to humans and animals globally. So for that, it's really important to control or to study novel strategies to control the transmission of these tick-borne pathogens. Recently, the study of the tick microbiota has become an tool for this approach as a strategy for a new control or new way to control these transmissions. The microbiota has been proven that play a role in the physiology, nutriology, and digestion, and is also playing a role in the transmission and colonization of pathogens. So, instead to ask, so what pathogens in this microbiota, we like to study how these microbes are interacting each other, and how this interaction could influence pathogen transmission. And microbial networks give us a really good approach for visualize this interaction that occur between or across the microbiota, the tick microbiota. In a graphical representation of a network, we have the nodes are representing are the microbes, and the edges between these microbes represent the interaction among those. That could be positive association or negative. This is a biological mean that gives to this positive interaction. We can say that is a cooperation, and this negative interaction that could be established in the network, we can say that is a competition. Not only for the visualization. Using a network approach, we can study how this community is assembled, we can identify keystone, and also we can evaluate how resilient is this community. For example of this, I want to show you some example of visualization of microbial network. Depending on what we want to show or what we want to express in our research, we can use different models to visualize. In the first case, I can show, for example, we can say larva, nymph, adult, how this microbiota change from the tick of the Rhipicephalus microbial from one stage to another. And on that case, we want to focus our visualization exclusively in the modules of the small communities that form the general tick microbiota. And we can see how this from a fragmented, more fragmented communities, they become a largest and more bigger module. In another type of visualization, we can represent only nodes. We can see in the right. We have nodes and connection. We can see how this connection or this interaction are dropping in one condition comparing to the other. More or not only visualization, we can also using this network approach to focus or target the exactly position of certain pathogen symbiote or bacteria in the community, and track how this target bacteria could move from one condition to another throughout life stage of under the infection condition. And specifically, we can see establish the subnetworks, a small the nodes that are directly correlated with our target bacteria or interesting bacteria, and create some network that we used to call in this kind of study local connectivity of our bacteria. It's important to take it in account that this is a tool, bioinformatical tool. We cannot do a really biological implication of what we are seeing here. This is just an interpretation and an easy way to visualize how the microbiota or the microbes that belong to the microbiota are interacted. Another important point that we evaluate using this structure of the microbial network is the selection of keystone. For us, a keystone are those bacteria that are present in all the conditions with high centrality and with high relative abundance. So using this approach, Mateos Hernandez et al. In 2020 identified an keystone, an Enterobacteriaceae, as keystone in the Ixodes ricinus microbiota. And using this approach, they developed an antimicrobiota vaccine that had been proved effect in the tip thickness and also in the transmission on the Borrelia axelis, blocking or reducing the number of spirochete, the total spirochete on the DNA. This same approach to study the centrality of certain taxa or microbes in the networks have been applied for other studies as Canos arguelles and Axeli in other systems, for example, for mosquitoes, to evaluate or to identify potential keystone that could be used as target or as antigen for the antimicrobiota vaccine and affect the fitness and competence or in different vectors. Last but not least, we have the robustness of the microbiota. That is the capacity of the resilience of the microbiota to resist external factors. So in this case, we have two approaches that we like to evaluate. The robustness we evaluate respect to a no addition or in terms of no removal. In this case, in the terms of the no removals, we select nodes with high centrality and we start to removing from the community, and we can evaluate how this community is dropping. And then we can evaluate in an already well-established community how the introduction of a new invasor, a new pathogen, a new bacteria could modulate, or if will be possible to modulate this community. One example of this we have it when we compare the microbiota of Rhipicephalus sanguineus and Rhipicephalus tyrannicus. We can see tyrannicus is the blue and the sanguineus is the red, and we can see that in the case of the tyrannicus, is required to remove less node to reach a loss of connectivity of 80%. So to reach at the fragmentation of that community compared to Rhipicephalus sanguineus. And in the case of addition, we have that they are lower average path length and higher largest component connected. That means that that network is more susceptible or permissive or easy to acquire new members in the community. All of this made the network at a easy tool to help us to visualize community organization and establish certain partners that could be, or interaction that could be hiding in the community to detect potential keystone species, and finally to see or to evaluate how that as strong as that community, how resilient is to external factors such as a dysbiosis that will cause the loss of member or the arrival of new pathogens. Thanks to all.
Thank you very much, Janet. A very clear presentation. So now is my turn. I will not expose anybody to problems with presentation because I do not have a presentation has a slide. So I will be speaking with you and transmitting a conceptual proposal. So the title of the talk is Re-Evaluating the Jenner-Pasteur Paradigm of Vaccination. But first of all, what are we understanding? Let's frame what do we understand by the Jenner-Pasteur paradigm, and why this idea of a paradigm has because I see this idea of a paradigm has a group of concepts, a group of conceptions that shape, that comes the important part, every imagination that we have about designing experiments, about designing vaccines. So it's a very important thing at some point in our research and our development as society to think what ideas are shaping our experimental program, our scientific questions themselves. So the Jenner paradigm refers precisely to how these core discoveries of Edward Jenner, so it was an English, let's say earlier researcher, and Louis Pasteur, a French researcher, as everybody may be aware, how those discoveries that they made shape how we design vaccines and how we conceptualize the design of a vaccine. And then, of course, how that paradigm fails to generate a protection against several pathogens, including vector-borne, and what could be the causes of that, and how vaccines of the future should include that criticism of the Jenner-Pasteur paradigm. So first of all, Jenner made a fundamental observation, Edward Jenner. He observed that people that were working with cows that have varicella, they didn't have human varicella. So exposure to the virus in the animal protect the other human animal against acquire a pathogen that was similar to the first one. That was a fundamental observation. However, it took several years for Louis Pasteur to make a jump in the understanding of that cross protection, because you were exposed to a pathogen from a cow and then be protected from a human pathogen. And then he using the virus of rabies, thought that maybe the same microbes being inactivated could protect against a future exposure against the same pathogen. And then he developed experiments in which he was the precursor of, say, vaccination, and he managed to protect against rabies virus. Those two sets of discoveries shape a conception of how shall we develop vaccination. So first of all, exposure to a pathogen or his components, because then later became recombinant vaccines, subunit vaccines, and triggers a protective immune response. So that's something we assume when we say we are creating a vaccine within that framework. Then this response, another assumption that we make, is predictable, specific, and durable in time. And then related to immune memory, exposure will develop protective immunity. Then that vaccination, which is another element, can mimic natural infection to create long-term immunity without causing disease. So then this, using that framework, we have been able to develop successful vaccines. For example, by successful vaccines, and I would be challenging that paradigm against eradication. So if we induce an immune response in a human population, as broader as can be, we could eradicate, for example, poliomyelitis in a full human population. It means that those concepts, exposing a certain population, the immune response of individuals in a population to antigens of poliomyelitis, and subsequent exposure blocks the transmission. So, or the transmission, no, the infection. So then you have, it's predictable, it's stable in time. So you have the boost could be for a long time to do reboost of the vaccine. So you keep this protection at the population level. However, when we go, for example, to malaria, to Plasmodium falciparum, the pathogen that causes human malaria, and we try to impose the same model. So then we start developing vaccines of inactivated Plasmodium falciparum, heat inactivated, chemically inactivated. We take subunits of the Plasmodium falciparum antigen and start in the search for immunity against Plasmodium falciparum using those tenets. We have a big failure. So today, I am by failure, I'm saying we have not arrived today, dedicating probably many more times resources and human minds involved in the development of vaccines against malaria, human malaria. We have not developed a vaccine as successful as we did, for example, for poliomyelitis. So in this moment, what's the question we need to ask? Haven't we found the right antigen? Haven't we discovered the right way to induce that immunity that we could induce in poliomyelitis? Or that path will never lead us to the development of a successful vaccine? Because malaria and Plasmodium falciparum may be playing, like other pathogens, by different rules that are not the ones that poliomyelitis. So that's the first proposal in the talk. What could be working in but it's the same, for example, the same. Let's go to another example, because that's vector-borne, but maybe also let's go to tuberculosis. So it has been very difficult to develop a vaccine that we could eradicate completely tuberculosis. And then we need to start then understanding, because then what, in my opinion, the Jenner-Pasteur paradigm has served also, I mean, the development of vaccines has a big ecological experiment. So by developing different vaccines against different pathogens, using that approach, we were able to identify across all these years of research what pathogens are really susceptible to having a population with high immunity against them, and then re-encounter will eliminate, and what pathogens ecologically, and the mechanism they have of evasion of immune response, of reservoir, etc., they will not be susceptible to be controlled, eradicated by increasing the immunity only in the humans that, or animals that would be exposed to the pathogen. So in the case of vector-borne diseases, which is following a bit the idea that Leonette was presenting, one of the problems, in my opinion, we could discuss after in the QA section, is that the boxes through which the pathogen move, they change. So you have a box, which is a vertebrate host, but then that box, the pathogen will not be forever there, so it will change to the other box that will be the vector. And that changes of the reservoir, so where the pathogen can persist in nature, that challenges the idea that I would raise a certain level of immunity in a population of humans and I will be able to eradicate Plasmodium. So we have to target, not only for vector-borne diseases, we have to target the vector, but targeting the vector itself, just because, of course, if we remove the vector for the formula, that's the easiest way to eradicate those diseases. So no vector, no transmission, but controlling the vectors. And I mean, in the third block of the webinar, we will be exploring those topics, has proven to be challenging. So a vaccine against mosquitoes, we are far away from a vaccine that we could protect against humans against mosquito bites. For ticks in animals, I think we have been more successful because of the feeding habits of the ticks compared to mosquitoes. And then you have vaccines that, for example, like Gavac, like subolecin based, that you could control the population of the vector in under certain conditions. It's not also easy because, for example, Ixodes ricinus being able to feed on across 200 species of birds, mammals, reptiles, is very difficult to develop a vaccine that you could control the vector itself. So it's challenging to just by raising immunity in the host, because actually the pathogen, let's go back to Plasmodium falciparum, have a mechanism to evade the immune response by, for example, antigenic drift, and then changing the surface, the antigenic surface of the pathogen, and then evading every time an immune response specific is developed, then the pathogen can switch to another antigenic variant and change, and so on and so forth. But then our proposal, and then coming back to the idea of building on what Leonette was discussing, recent years, in the last 30 years, and also what Salma tried to convey in her presentation, we have accumulated strong evidence that the microbiome of vectors have very strong and very intimate interaction with the pathogens that they carry and they transmit. And you can imagine that there'll be a magic there. So we have microorganisms, and then you have competition. So you have a community of bacteria, of fungi, of viruses within the vector, and then you have something new entering, has a secondary visitor. Let's say that's the pathogen always arrives later to the party, unless you have a pathogen maybe that is transovarially transmitted, and they will be maybe established very early in the microbial community of the vector. But otherwise, the pathogen that doesn't have a transovarial transmission, vertical transmission, and is horizontally transmitted, they would be all the time arriving to a microbial community that would be established. That would create barriers for that establishment of the pathogen. So there is a lot of literature on that event or phenomena that is called colonization resistance. So the arrival of a new member of that community would face resistance for current residents in that community. So the pathogen will face that, and in that struggle, sometimes it will succeed to colonize the vector, and sometimes it will fail. So there is evidence showing that the composition of the microbiome, I saw a question somewhere in the QA that was asking whether there is evidence. We could discuss after in the question, but there is evidence showing that the composition of the microbiome of the vector before pathogen arrival can shape the colonization, the capacity of that pathogen to colonize the vector, and then to be transmitted. So from that struggle, it's not obvious that the pathogen will colonize. And in fact, when you test pathogens in vectors feeding, for example, we did a very beautiful experiment on cows. So we were ticks feeding on cows, and the cows were, as they become chronically infected with Anaplasma marginale. And then we test the ticks feeding on the cow against Anaplasma marginale. Not 100% of the ticks feeding on the cow, chronically infected with Anaplasma marginale, are infected with the same pathogen. So exposure, and we are talking about Rhipicephalus microplus, so one host feeding on the same cattle, and then after having different stage, adult stage, some of them were not infected with Anaplasma marginale. So you have changes in that capacity. Of course, there would be maybe also, I would not disregard the other elements like genetics of the vector, maybe other factors, immunity of the vectors, that may be also influencing how the pathogen is not entering. But the microbiome is also playing an important role in shaping the vector immune response itself. So changing in the composition of the microbiome of the vector could shape the immune response of the vector against the pathogen, posing challenges for the pathogen to enter due to colonization resistant mechanism, and so on and so forth. So the idea here, going back to the idea of vaccination and how we challenge that paradigm of Jenner Pasteur in the case of vector-borne, our proposal is that we try to develop strategies that do not target the pathogen whatsoever. So we are not interested now in that Pasteur-Jenner tenet that is a specific immunity against the pathogen will protect against the pathogen. So we want to target the microbiota of the vector. So how can we, using antibodies as a way to deliver means of manipulation of the microbiome of the vector, how using those antibodies that target a bacteria that is in the microbiota of the vector that have no relation whatsoever with the pathogen we are interested in controlling, we could induce certain changes in the microbiome that then the vector become non-permissible for the infection with the pathogen. So if we manage to address that problem, departing from the tenets of the Jenner Pasteur, and let's keep fighting for finding the right antigen, we could maybe track the pathogen in the other box. So then we move also from controlling the pathogen within the box of the vertebrate host to the box of the vector, because now we could track with the antibodies that has been shown that they can be stable across time within the vectors. In ticks, even some of the antibodies escape the proteolytic machinery of digestion of the tick, and they get shuttled back to the host. So they go through the intestinal tract, they go to the hemolines, salivary glands, and they re-enter the host. So you could see that there is a certain mechanism that some of this reshuttle of the antibody has been hypothesized that may be one of the mechanisms by which the vector uses to manipulate the immune response of the host. That's a different issue, because the other day talking to Ana Laura, she's going to present, she brought that because I was discussing with her a bit the topic I wanted to bring into the talk today. And there's also another element of the saliva of the vector and the immune modulation of the vector of the immune response of the host itself, so that the vector is not a syringe. That has been shown by several studies with Labuda, Patricia Nuttall. They showed that the syringe inoculation of the pathogen, vector-borne, is not as efficient as when we associate it with the saliva of the vector. So the vector has different functions assisting the pathogen. But our proposal, and then I will be shaping the conclusion and the closing statement, is to devise strategies by which we could modulate the vector microbiome through vaccination. It is one approach to stay within the concept of vaccination, but we could also develop other strategies like paratransgenesis, or it is known with Wolbachia in mosquitoes that they have been success on that. So challenging the current understandings we have and the immunological school, because I think there is a strong influence from the the immunologists, how to say, gremio of immunologists, that the immunologists are obsessed with the specificity. And that brings a lot of constraint and let's say a bias when we are talking about designing a vaccine. So, let's design a vaccine against Lyme disease, and let's have, and we vaccinate the host against Escherichia coli. Okay, the immunologist will not pass that grant, so probably you will not get the money that you need to do that research. So, but questioning that core conceptions, we could develop new vaccines of a new generation, and maybe we could reach that goal of eradication of vector-borne pathogens by departing from that paradigm. So with that statement, I close my presentation, and then we have 10 minutes for QA section. Alexander, would you help us there so you could address the or comments? If somebody have comments, can you want to refuse what we just said or accept it or whatever is open for discussion? It would be great to hear you guys.
Yeah, thank you, Dr. Cruz. Now we will have 10 minutes for Q&A session. There is just one written question. I believe you already commented it, but if you want to add something, you can check the text box of Q&A section if you wish to add something. And of course, maybe there is somebody who would like to ask something live verbally. So please go forward with your questions.
Yes. So we have a question. They say, are there lab experiments on changes in bacterial competence in relation to microbiome composition? So that was, there were, for example, in ticks, just to point out, because before I make a general statement, in ticks, there is what I consider is a beautiful experiment by the group of Eero Fikrik, that in which they showed that ticks raised on an sterile environment in the lab, I mean, more sterile than the lab itself, more clean. So they have a different capacity to acquire Borrelia burgdorferi. So just by being raised, and that they show in that experiment that Borrelia, that the microbiome changes when the ticks are raised in an environment that is more sterile, and the ticks were the same. So here there is a certain control for the genetic of the ticks and so on and so forth. So an experiment that puts other factors apart. So the microbiome only, let's just change nothing, not the big biological changes, the sterility and not the sterility, and you have a change in the capacity of that tick to acquire and the of Borrelia to colonize that vector. So that's one of the experiments in ticks that I am aware of, and that shows is an example of that. But having found that example, you will not find strange that this is the only example. So there would be many examples of the same. So I don't know if that answered the question. Ana Paola was the one asking if it's okay, then we move, or is not, please.
We will wait. Maybe she will add something in the chat. I see that
I think Alina want to say.
Alina raised the hand.
Good morning, Alan. Very interesting. I would like to ask you about your point of view about the future of this approach to control pathogens transmitted by ticks, because as you say, this is a complex system, and using vaccines, you are able to modify some microorganisms that are a positive relationship with the pathogen that you want to modify. But in the case that you have a negative relationship, that is not possible to use vaccines. You think that the future approach will be a combination between vaccination and other modification because I think that modification of only one pathogen difficult will be elimination of the pathogen that you want to control. In this case, I think it's more it's better to use a comprehensive approach. And for this reason, what is your comment about that?
Alina, I thought we were on the same team. It's like that's a joke. It's a joke. No, yes. Yeah. No, that's a very good question. I think one approach, I think one conception, and we are been defending this concept. One approach is precisely finding the right candidate on the microbiome based on the suggestion you just made, which is find a bacteria that have a positive association with a certain pathogen of interest, and then you removing that member, you hypothesize that you have, and that's a bit of a very positive approach, rational approach to discover the target. However, we are proposing a different approach in which what we do is by targeting a keystone taxa. So a taxa that is a central taxon for the structuration of the community of the microbiome, a central taxon for the community of the microbiome, and may not have links, direct links with the pathogen itself. Targeting the taxa, you could modulate the microbiome, the whole shape of the microbiome. In the understanding microbiome, in the way Lianet was explaining, has a network of interaction that if you change one of the elements that have positive interaction with this one, negative with this one, positive with this one, so it's a domino effect that then you modulate the full community. That full community modulation could render a infection refractory state within that microbiome. So now it's about a global approach, but then I understand that this one is not very appealing because how you rationalize what that state looks like and how I can repeat that state across vectors. So it's a complicated prediction to make. Your approach is more rational and more predictable. So I target this one is positive with this, and then if I remove this one goes down. So definitively more research is needed on that direction, but we are proposing these two approaches to find, to apply this type of vaccine.
Thank you for the comment.
No, thanks to you.
Thank you. There are two more questions in the Q&A section, so you can check.
Yes. Yeah, Omer, thank us for the information. Thank you, Omer. And then Ludovica have a question for Salma. Salma, are you around?
Yes, I'm here.
So Ludovica is asking when and where the hyalomatics were collected, from which host, if there was an animal host collected.
They were collected from cattle for 4 seasons for pathogens screening, and for 3 seasons for microbiome analysis.
Yes. Well, Ludovica, if that information answer your question, perfect. If not, Salma would be very happy to catch up after if you drop an email or something. Thank you, Salma. So an anonymous attendee have a question. Thank you for your comment. So is there any information about if a significant difference of microbiome in ticks that have more than one host exists? And does it may also change the capacity for acquiring pathogenic strains when passing from one host? Yeah, but that's it. That's actually one of the points. First of all, it has been shown, yes, the host, the type of host the ticks feed on will modulate the microbiome in a different way. So the tick feeding on the lizard will have a modulation of the microbiome different on a tick feeding on a cow or a tick feeding on a dog. So here it's not mystery about how could that be. First, the element of rationality there would be that each of those hosts themselves, they have a very specific microbiome, and the tick being crawling on the skin and being exposed to the microbiome of that host. So you can imagine that it would be acquiring different bacteria from each of the hosts they are feeding on, and the acquisition of those bacteria will shape the microbiome that the tick has previous to the feeding in a very different ways compared to host. So the question is, yes, And the question that would be very interesting to maybe to assess. For example, Borrelia would be a very interesting model to test that hypothesis you say. So if I have a lizard, a rabbit, and a dog infected with Borrelia, and I put the tick to acquire Borrelia from the different hosts, a tick that is genetically similar from the same mothers and so on and so forth, would Borrelia will enter to the tick in different proportions based on that modulation difference that the host itself induces. So that would be a beautiful experiment to do, my friend, whoever you are, the anonymous attendee. So I hope we reply all the questions. So maybe, Alexander, I know what you think. We can move to the next block.
Yes, I agree. We can move to the next block. So the next speaker will be Miss Dagmara, and so I invite her to share her screen.
Okay, so thank you very much for inviting me. And today I will be talking about actually my PhD project, which is about lesser known tick species around the world. So as we all know, ticks transmit the widest range of pathogens globally, and recent climate shifts and human-induced habitat changes have facilitated the spread of tick into new geographic areas, introducing emerging diseases to previously unaffected populations. And the ecological significance is rising due to the increased environmental suitability in temperature zones, greater contact with humans and domestic animals, and co-infectors which may worsen the disease. But there's a lot of studies about ticks and tick-borne diseases around the world, but we noticed that there is two actually research gaps, like a great majority of review studies on Babesia were performed on the most common European tick species, Ixodes ricinus and Dermacentor reticulatus, but also a marked number of studies was conducted on Ixodes scapularis. But the other important tick species like Hyaloma and Hemophysalis ambioma and Rhipicephalus were rare or performed on ticks collected from domestic animals or livestock. And studies on partially engorged feeding ticks can lead to misleading conclusions, as the detection of the genetic material of pathogens in such ticks may be a result of host infection and milk contamination in ticks. So why our project is so important? Firstly, because ticks are the most dangerous vectors of pathogens in northern Hampshire, and they transmitting numerous bacteria, viruses, parasites such as Babesia and Theileria. And 16 tick-borne diseases have already been described in humans, 4 of which were only detected after 2013. And the risk of infection also depends on the tick species, its density in the field, and the frequency with which is infected with the microorganism in questions. And also climate warming further increase this risk. And exotic species are also emerging in Europe, like Hyaloma and Hemophysalis, which can establish themselves and introduce new pathogens in an increasingly warm environment, although actually knowledge of their epidemiological role is still limited. So the main goal of our project is actually a global study on Babesia and Theileria occurrence in the most important tick species from 4 continents, which is Asia, Africa, Southern and Northern Americas. And additionally, Borrelia burgdorferi infection in different tick species will be assessed. And here are our basic hypotheses. Like first, we hypothesize that there's high specificity in Babesia Theileria ticks interactions, that co-infections with different Babesia can occur in ticks worldwide, and that there's a specificity in Borrelia tick interactions. So it's still an ongoing project. We're still on the beginning, but we have a few up-to-date results. So first, I will talk about the results from Mongolia. So we conducted 2 field expeditions, one in 2023 and the second in 2024, and we collected the ticks from different biotopes. It was questing ticks. We collected the ticks on the taiga, in the steppes, and on the deserts. And during these 2 field expeditions, we collected 3 tick species. It was Ixodes persulcatus, Hyaloma asiaticum, and Dermacentor nutari. So actually, the highest level of infection for Borrelia and for Babesia we discovered in Ixodes persulatus, and we discovered Borrelia bavariensis, Babesia cabali, Babesia venatorum, and Babesia microti in the sticks. In the case of Dermacentur natali, we detected Borrelia avzeni and Babesia cabali, but the infection occurred actually in a very low level. And in the case of Hyaloma asiaticum, despite the high number of collected Hyaloma asiaticum questing ticks, we didn't discover any Borrelia and any Babesia in the sticks, but we still have 500 ticks from animals, which we can study and see if we can detect some Borrelia and Babesia in the sticks. And here we have the phylogenetic trees for Borrelia. So actually, this tree reveals two distinct Borrelia bavariensis clades, and Asian clade and European clade. And this Borrelia bavariensis we discovered clustered together with the other strains from Iran and Russia. And this still required further analysis. And Borrelia avzeri clustered together with other Borrelia avzeri isolated from Ixodes persulatus in Western Siberia and from Ixodes ricinus from Poland. And here we have the phylogenetic analysis for Babesia. So what's interesting, that Babesia cabali splits on this tree on 3 distinct clades. And Babesia cabali isolated from Ixodes persulatus and Dermacentur natali clustered together with strains from China and Japan. And Babesia venatorum clustered with Babesia venatorum isolated from ticks and animals in Czech Republic and France. And we also collected few ticks in our country. In Poland, we collected 200 Ixodes ricinus ticks and 200 Dermacentur reticulatus ticks. And Ixodes ricinus show a high infection rate for Borrelia and Babesia. We detected Borrelia avzeli, Borrelia garini, Babesia microti, and Babesia venatorum in the sticks. And when it comes to Dermacentur reticulatus, we have 1 tick positive for Babesia canis and 2 ticks positive for Borrelia avzeli. And here we also have the phylogenetic analysis. So when we look on the Borrelia, we see that Borrelia avzeli and Borrelia garini clustered with other strains isolated from ticks and animals from Poland. And when it comes to Babesia, for example, Babesia canis, we can see that it clustered with other Babesia isolated from animals and from ticks around Europe. And we have the similar situation for Babesia microti and Babesia venatorum. And we also, due to our collaboration, we received a few ticks from Romania. It was Rhipicephalus rotilius, and 2 ticks were positive for Babesia vogeli. And this Babesia vogeli resulted in 100% identity to Babesia vogeli isolated from ticks and animals in Romania, Egypt, and Kyrgyzstan. And we also conducted studies in Brazil, but we still study these ticks. We collected the ticks in Maranhão, and we collected the ticks in 9 different locations. It was Forest River area, meadows and grasslands, farms, and burn areas with ant nest. We also collected the ticks from dogs, livestock, and wildlife. We collected all tick stages. And to sum up, we collected above 1,200 ticks. Most of them were Ambioma sculptum, but we also collected Rhipicephalus sanguineus, Rhipicephalus linnei, Ambioma caenesis, and Dermacentur nites from horses. But we still study this tick. So to sum up this project, we actually plan to at least study 8,000 ticks from different genera. This February, we also have been to Africa and collected the ticks from Zambia and Ethiopia. We collected above 3,000 ticks, which now are we now identify by morphologically, and this next month we also plan to come back to Africa, to the other countries like Namibia, Botswana, and Zimbabwe to collect more ticks. And we also plan this October go to USA for more tick collections. And thank you so much for your attention. I also want to thank my supervisor, Professor Anna Bayer, for her outstanding role as supervisor. And as I said, it's still an ongoing project and we still have a lot of things to study and a lot of things to discover. So thank you very much for your attention.
Thank you, Dagmara, for your presentation. And now we can proceed with next speaker. So I invite Dr. Kivo to share his screen.
Okay, thank you very much. So welcome everybody. Good morning, good afternoon, or good evening, depending on where you are on the globe now. My name is Gergő Keve, and I'm a veterinarian from Hungary. And today I'm going to speak about the role of birds in dispersal of ticks and other arthropod vectors, or actually some aspect of this quite huge topic, actually. So it's long since known that birds can carry a lot of ticks to great distances. Actually, from a European point of view, the most important are the spring migrants that can carry hyalomas or even amblyoma ticks from Africa to Europe. On the other hand, the autumn migrants can be of great importance as they play a great part in the distribution of the local tick fauna. So let's just see what we did here in Hungary. So during the birds ringing, ticks and other arthropod vectors were collected from the avian hosts. This was conducted at 7 locations of Hungary. Óca in central Hungary, a city, deserves extra attention as at that place, the sample collection started at the March of 2015, while the other location signed with green joined at 2022. So when after the samples arrived to our laboratory, we checked them morphologically and also conducted molecular analysis as well as phylogenetic and statistical analysis. As per the results, I just wanted to speak about 2 manuscripts of ours that are already published. The first one is published in Scientific Reports. It is about the long-term evaluation, so the temporal evaluation of tick distribution in Hungary. It is based on an 8-year-old study conducted in central Hungary in Óca. During this study, we collected 10 different species of ticks from multiple birds. As you see here, Ixodes ricinus predominated in our sample collection. Actually, this is nothing new, as in Central and Northern Europe, Ixodes ricinus is known to be the most common ticks found on birds. But the second was Haemaphysalis concinna, which is, well, accidentally, it is a continuation of the previous presentation. Haemaphysalis concinna is a lesser known tick, so I will just focus on this one. Haemaphysalis concinna, interestingly, it is not that common in Europe. However, in the Carpathian Basin, so in Hungary, for example, it has a hotspot. It is much more prevalent in this area than elsewhere. That is why we can actually work with this. On this WISP-web, I tried to summarize the tick-host interactions that we observed. And it was striking that Locustella luscinioides, so the Savis warbler, was by far the most common host of Haemaphysalis concinna. In the case of Ixodes ricinus, Turdus merula and Erytacus rubeculus, so the blackbird and the European robin were the most common. However, this is again nothing new. This is similar in many other European countries. So let's just focus on Savis warbler, this beautiful brown reed-associated long migrant bird, which is by far the most common avian host of Haemaphysalis concinna, at least in Hungary. Actually, on this table, I listed 5 most common hosts of avian hosts of Haemaphysalis concinna we found, and this is striking that Savis warbler was responsible for 55% of the total Haemaphysalis concinna we found during the 8-year-long study. Parallel to that, the other warblers of different genus, so Acrocephalus species, contributed to this as well. Together, the 3 warbler species carried 72% of the total Haemaphysalis concinna collection we found. So based on this, we decided to compare the hosts of Ixodes ricinus and Hemaphysalis concina, the two most abundant species during our study. So based on the host attributes, we first examined the habitat of the hosts. There was a significant difference between the two species, where Ixodes ricinus was the most common on forest or meadow habitat birds, while Hemaphysalis concina was by far the most common on reed-associated birds, and especially those three warblers I mentioned before. The migration habit was also examined. Ixodes ricinus was by far the most common on resident and short-distance migrant birds, so the local birds. On the other hand, Hemaphysalis concina was much more prevalent on long-distance migrants. The feeding place of the hosts of these ticks were also analyzed, and Ixodes ricinus found to be more common on ground feeding birds, while Hemaphysalis concina is on above ground feeding birds. So about the temporal distribution, since there was no real significant difference between each year, I decided to show you the data from 2015. So as it's visible here, during the spring migration, the nymphs of Ixodes ricinus predominated, while on the other hand, the larvae were the most abundant during the autumn migration and the end of the nesting season. In the case of Hemaphysalis concina, the situation is a little bit more complicated, as Hemaphysalis concina is a thermophilic tick species. So it was expected that we found it during the summer months. However, they were by far the most abundant during July and August, which is in fact the most active period of the most common hosts. So there might be some bias introduced by these three avian host species that I introduced before. We also conducted analysis temporal distribution of Ixodes frontalis, which is an ornithophilic tick species. However, this is, in contrary to Hemaphysalis concina, is a tick that is very common in the winter months. So our sample collection basically started in March and ended in the end of October each year. Therefore, it is not surprising that we found the most Ixodes frontalis in the beginning of our sample collection, but from the winter months, we had only sporadic data. So the other results that I wanted to speak about concentrates on the spatial distribution of ticks in Hungary, which was conducted in 2022. At 7 different locations in Hungary, we collected ticks from birds continuously from March to November. And during this, we found something much more interesting than just Hemaphysalis concina and Ixodes ricinus, and that is Hyalomma rufipes. Now, actually, Hyalomma rufipes is not uncommon on birds in Europe. This was found even in Norway, even on Finland on migrant birds. What was striking, however, that 7 of the 12 specimens we found were feeding on Pandurus biarmicus, or bearded reedling, which is a sedentary or resident bird species. So we found an African tick on a resident bird that never left Europe. Now, 7 of these ticks were found on Pandurus biarmicus, one of them being a larva, which was unfed, by the way. At the same location where we found the ticks on the Pandurus biarmicus, we found 9 nymphs and 1 larva in total. And all of them were found on the June 16th of 2022, which is in the breeding season of the other species, the sedge warbler. We conducted molecular analysis on these ticks, and based on the 16S rRNA, we found that those we found at the same place differed to the other 2 nymphs we found elsewhere, but they belong into the same genetic background. So, a discussion regarding this. So what we think we found is a reproduction of Hyalomma rufipes under European circumstances. This was actually foreshadowed by many authors before, and some authors from the previous century actually suggested that this has already happened in Europe before. Prior to our study, research from Czechia underlined that the Hylomarufipes may be able to overwinter in Central European conditions. So what we think happened is the accidental finding of a male and a female each other, and they were able to reproduce. What we found may be an opportunistic population. Of course, this population needed to be assessed whether they can survive the Hungarian conditions or not. For that reason, we conducted a large-scale sample collection around the suspected population. The black cross here shows where it happened. And around it, we conducted a lot of sample collections with the help of veterinarians in the area. But despite our efforts, we found 0 Hylomarufipes adults after we discovered the population, which means that the population may not be able to survive. But nevertheless, it was the first report of reproduction of Hylomarufipes under European circumstances, but it was not able to survive. So before finishing, I just wanted to mention that it's not just ticks we examine here from birds, but other parasites. For example, louse flies, which are also significant parasites of birds. This specimen here is Ornithotonor laticornis, which is just like Hylomarufipes, a sub-Saharan species, which was first discovered in Hungary during our research and first time in Europe. So thank you very much for your attention, and special thanks to my co-authors. And thank you very much for your attention again.
Thank you very much to you also, Dr. Keve, for your presentation. And then I will invite next speaker, Professor Dr. Marco Andre. So you can share your screen now.
Thank you very much for your presence today. And I'm gonna talk a little bit about the diversity of paraplasmids in wild animals from Brazil. So back in 2019, Yellow and colleagues have established the presence of 10 paraplasmid clades, which was named from 1 to 10. And today, I'm gonna try to show you guys what we have been done regarding the expansion of this paraplasmid clade. So Yellow have classified the paraplasmid clades into Babesia sensu stricto, Theileria sensu stricto, Theileria equus group, Cyrosum, Rhinoceratid group, Marsupiala group, Pierce, Western group, Monotremata group, and the Babesia microtylite group, which represents the clade 1. So they observed that most paraplasmid lineages were associated within the respective vertebrate host taxon, suggesting then that paraplasmid lineages have principally evolved by co-speciation with their mammalian and or bird host taxons. Let's start our journey by talking about opossums. So we have several species of opossums in Brazil, and we try to seek if opossums were parasitized by Babesia or Theileria species. Then we collected blood samples from Gidelphis albiventris, which is this guy, from central western Brazil. And during the examination of blood smears, you guys can see here that we found trophozoites inside the red blood cells of these guys. So then we collected blood samples from Gidelphis aurita in Rio de Janeiro. And we came across trophozoites, but this is really interesting because we found a paraplasmid which resemble the Maltese cross from Theileria. So when we performed a phylogenetic tree based on the near complete 18S RNA, what we saw, we detected a new phylogenetic clade, which we called South American marsupial clade, which encompasses the paraplasmid that we detected in the Delphis aurita from Rio de Janeiro, as well as the paraplasmid which we detected in the Delphis albivrentis from central western Brazil. Then we decided to perform another phylogenetic tree based on a mitochondrial gene called the COX-1. And again, we can see here the uniqueness of these phylogenetic trees, which comprise only paraplasmid from opossums from Brazil so far. Then we went to northeastern Brazil in the state of Maranhão, and we collected blood samples from the Delphis marsupialis, which is the third the Delphis species found in Brazil. And again, you guys can see here the uniqueness of this highly supported clade, which comprises Babesia from the Delphis species from Brazil. So what about the tick vector of this putative novel paraplasmid? So we suspected that Amblyomma dubitatum, which is a tick species very commonly found on opossums, can vector these new paraplasmids. Why? First of all, because we detected these paraplasmids in Amblyomma dubitatum ticks collected from opossums, but we also detected these paraplasmids in Amblyomma dubitatum ticks collected from Nasua quachis, which are not hosts for this putative novel paraplasmid. So let's jump to bats. So we have a huge diversity of bats in Brazil. We have around 186 bat species in Brazil. So these bat species can harbor Babesia or Theileria. So in 2021, we detected a putative, again, novel paraplasmid species in bats from central western Brazil. So we collected blood samples from bats in the Brazilian savanna in the state of Mato Grosso do Sul, which is located in central western Brazil. And what about the phylogenetic position of this paraplasmid from bats? Right here, you guys can see that, again, we have another and distinct phylogenetic clade that comprises only paraplasmid that we detected in Philostomum discolor from central western Brazil. So we have added more two phylogenetic clades to those that have been previously supported by Yellow-Veca. What about tapirs? So we have a huge diversity of wild animals in Brazil, and we presume that we also have, because of that, a huge diversity of paraplasmids. So there were a couple of previous studies in Brazil based on short fragments of 18S RNA that have claimed that tapirs could be hosts for Theileria equi, but we would like to look into more deeply on that. So we collected blood samples from tapirs from two different biomes in Brazil, namely Cerrado, which is similar to Africa savanna, and the Pantanal, which is the largest wetland in South America. And we detected in blood smears from the lowland tapirs a Theilerid species, you guys can see here, that was smaller than Theileria equi from horses. So we found a prevalence, a molecular prevalence of more than 50% of positive for paraplasmids in tapirs from the states of Mato Grosso do Sul. But what about the phylogenetic position of this paraplasmid species from tapirs? So right here, you guys can see here that the tailaria that we have been found in tapirs were closely related, albeit very distinctive from Tailaria equi. So we named this new species as Tailaria terrestris. So there is a very well supported clade that separated the Tapirus terrestris clade from Tailaria equi and the Tailaria honey. And this was based on the near complete 18S RNA. What about the topology when it comes to a COX-1, which is mitochondrial gene? So right here, again, you can see that very high supported. There is a separation between the Tapirus terrestris clade and the ECOS group, which has been shown by Jaloveca and colleagues. And these results were also supported when we performed a phylogenetic tree based on the heat shock protein 70. Again, very closely related to Tailaria equi, but distinct from it. What about capybaras? So let's jump to rodents. So capybaras are larger rodents that has a huge importance in Brazil because they are amplifiers for Rickettsia rickettsii, which is the causative agent of the Brazilian spotted fever. So in a collaboration with other colleagues from Central Western Brazil, we detected a babesia, a large babesia in blood smears from capybaras. What about the phylogenetic position of this putative novel babesia? So again, we discovered another phylogenetic clade, which was distinct from other phylogenetic clades that has been shown before. So we add a novel phylogenetic clade. We called it capybara group, and it is very distinct from all other babesia clades. And we also have shown based on other molecular markers, the very distinct phylogenetic clade, and we called this babesia, we named it as Babesia goianiensis. This is the HSP70 based phylogenetic tree, and this is based on COX-3. All topologies were supported by each other. What about the tick vectors of Babesia goianiensis? So we collected blood samples from Coatis in Midwestern Brazil, and we detected Babesia goianiensis in Amblyoma sculptum ticks, which were collected from Nasua in Campo Grande. So Coatis were negative for this babesia, so we presume that these ticks have fed on capybaras before jumping into Coatis. What about wild rodents? So this year we published this paper, and we described another phylogenetic clade. We called it a South America rodentia group. We collected blood samples and the tissue samples from rodents in Mato Grosso state, which is located in central western Brazil. Again, we detected another phylogenetic clade. We call it South American rodentia group, which is closely related but distinct from paraplasmid clade that we have described in bats from central western Brazil. And these results were supported by another phylogenetic tree based on COX gene. What about Babesia species in wild canids in Brazil? So we described a novel Babesia. We call it Babesia pantanalensis because it was detected in crab eating fox from the Brazilian Pantanal. And we detected this novel Babesia in 3 out of 12 crab eating fox in the Brazilian Pantanal of states of Mato Grosso do Sul. So wild canids in Brazil are hosts for another paraplasm that only occurs in South America. It's called Rangelia vitalii, which is a Babesia sensu stricto, but different from other Babesia species. This parasite can infect it. Besides these red blood cells, it can infect leukocytes and endothelial cells. And different from Babesia vogeli, which is less pathogenic, Rangelia vitalii is very aggressive to dogs, and it can cause a very severe disease with several hemorrhages in guts, eyes, and the several parts of the body. The vector is Amblyoma areolatum, which occurs in southeastern and South Brazil. And Cerdocyon the crab-eating fox, are considered the most common hosts for both Rangelia vitalii and the ticks of the species Amblyoma areolatum. So what we have found in crab-eating fox from the Brazilian Pantanal. So in the Brazilian Pantanal, we do not find Amblyoma areolatum. So we do not expect to find the Rangelia vitalii, okay? We detected a Babesia, which was very closely related to Rangelia vitalii, but distinct from it by high bootstrap value. So this is the phylogenetic tree based on the near complete 18S RNA, but we had to show it based on other distinct molecular markers. So based on HSP70, we show again Babesia pantanalensis here, which shown to be, again, closely related to Rangelia vitalii and to Babesia coco. Babesia coco is a Babesia that has been detected in dogs from North Carolina states, the United States, sorry. And we shown, again, based on COX-1 gene, that Babesia pantanalensis, again, is closely related to Babesia coco and the Rangelia vitalii. So based on distinct molecular markers, we named it as a novel species of Babesia in an area where we cannot find Amblyoma areolatum, which is the vector for Rangelia vitalii. What about the suspected vector? So we suspected that Amblyoma parvum is the vector of this novel Babesia. Why? Because 10 years ago, we performed a study in the Brazilian Pantanal, and we collected ticks from wild rodents. In those ticks, we detect the same Babesia, but by that time, we only got a short fragment of 18S RNA, and we were like, oh, there is a Babesia which is closely related to Rangelia vitalii, but we don't have the tick species here. So we called it by that time as Babesia SP. So rodents are not hosts so far for this novel Babesia. To finish, what about wild felids? We have a huge diversity of wild felids in Brazil. And as you guys may know, wild felids are the main hosts for cytozoom. So several papers in Brazil have shown that wild felids in Brazil are the main hosts for cytosol. But with cytosol, when we seek to sequence very short fragments of 18S RNA, this cytosol was closely related to cytosol philis, which occurs only so far in the USA. So last year, we described a new cytosol called Cytosol brasiliensis in a little spotted cat in central western Brazil, in the city of Brasilia, which is the capital of Brazil. So right here, you guys can see the merozoites, which are very pleomorphic, and the schizonts in the liver. Regarding the phylogenetic tree, you guys can see here that based on the near complete 18S RNA, we have the clades of Cytosol philis and the Brazilian clade, which comprises Cytosol brasiliensis. So these results were supported by the phylogenetic tree based on the near complete cytochrome B, which is a mitochondrial gene. And again, we can see here that Cytosol brasiliensis is very closely related, but distinct from Cytosol fellis. Okay, this was supported by a high bootstrap value. So we try to expand our knowledge on this diversity of Cytosol in jaguars from Brazil. So jaguars are very beautiful wild felids, and these are ocelots. So we got several collaborations and got blood samples from jaguars from several Brazilian regions, and from Leopardus pardalis from Mato Grosso do Sul, Puma concolor, and Leopardus bragatus. What we discovered? So we detected Cytosol brasiliensis in Leopardus pardalis, but not in jaguars, okay? And the jaguars were indeed parasitized by several genovariants of Cytosol philis. So so far in Brazil, we have to say that we have at least 2 Cytosol in wild felids from Brazil. So Cytosol philis and the Cytosol brasiliensis. So this is the near complete 18S based phylogenetic tree. This is the cytochrome B based phylogenetic tree, and they were supported by each other and by high supported bootstraps. These results were confirmed by COX-1, which is a mitochondrial gene. So Cytosol philis, several genovariants in Panthera onca, which are jaguars, and Cytosol brasiliensis in ocelots. When we run a phylogenetic tree based on a more polymorphic intergenic region, ITS1 and ITS2, we found the same results, showing that Leopardus pardalis were parasitized by Cytosol brasiliensis. And we also detected a sequence from a cat, which is supposed to be Cytosol brasiliensis and not Cytosol philis. That's why we do not see cats dying of cytozoonosis in Brazil, which is very common in the USA. So probably because cats are infected by Cytosol brasiliensis. What about the vector? Just to finish this presentation, we suspected that Amblyomys cuptum is the main vector of Cytosol among wild felids in Brazil. Why is that? Because 3 years ago, we collected Cytosol, we collected ticks from wild boars in Brazil, and we detect Cytosol in those ticks. Wild boars are not hosts for Cytosol. So we presume that Those ticks fed previously on cats or wild cats, and then jumped to wild boars. And in the states where we cannot find the amblyomys sculptum, wild felids are not positive for cytosine. So in those states where you can find cytosine, you can find amblyomys sculptum. But in those states where there is no amblyomys sculptum, all wild felids and cats were negative for cytosine. So in a nutshell, we have expanded the university of the paraplasmid phylogenetic clades. So from 10 phylogenetic clades, we expanded them to 15 based on the work that we have been done in the last 5 years. So taking home message, I very like this paper from James Harris that he says, taxonomy should reflect phylogeny and recent advance in this field, especially the use of molecular techniques, which has led to many reclassification. However, such taxonomic changes must follow the zoological nomenclature codes. And finally, we strongly suggest the use of less formal terms such as South American marsupial group, South America rodentia group, capybara group, in order to avoid taxonomic instability by employing inappropriate scientific names until sufficient data allow the formal description of species and place them within a taxonomic context. I'd like to thank FAPESP and CNPq for the financial support and all my team for the hard work. Thank you very much for your attention. I would be more than happy to take questions. Sorry.
So thank you, Dr. Marcos, and thank you for your presentation. It is totally fine. Next session is about Q&A. So I will check just if we have some questions left by attendees, but so far I think there are no questions. I will wait for a minute to see if anybody will leave the question. If not, then we can proceed with the next group of speakers. So I will please all attendees, if you have any questions, please leave them in the Q&A box. Maybe later somebody will leave the question at the end of the webinar, so maybe we can later check again.
Okay.
During the last Q&A session.
Okay.
Okay, now I will invite Ana Laura, Dr. Ana Laura, for her presentation. So please proceed with the screen share.
I cannot do it. I need the Professor Marco stop sharing.
So, Professor Marco, can you check and stop sharing screen?
Okay, sorry. Sometimes I have difficulties with this technology.
Totally fine. We will wait.
Where should I?
You should see something like a little red mark where it's written.
Okay.
Stop sharing.
I see it now. Sorry.
Okay,
now it's fine. So, Dr. Ana Laura, you can try now.
Like this, just in case I cannot put it on full screen. So, well, I would like to thank to Alina and Alejandro for the invitation to this webinar, for the opportunity to share the research of our lab. Today, I will talk about the work of our lab has been doing in the last few years, focus on different strategies to develop anti-tick vaccine against Ornithodoros of ticks. First, I will introduce the tick species that we work with, Ornithodoros erraticus and Ornithodoros movata. Ornithodoros erraticus is distributed across the Mediterranean Basin, the Middle East and part of Asia. In the case of Ornithodorus mubata, is mainly present in the southern east of Africa mainland and Madagascar. This tick species belongs to the Argacidae family, also known as soft ticks, and they have an endophilic lifestyle and they can feed in less than an hour. Why are these ticks species are important? Well, they are vectors from for several diseases such as African swine fever, which affects domestic pigs and can have a high mortality rate, reaching 100% in some cases. But they also can transmit other Borrelia spirochetes, such as these ones, and causing tick-borne relapsing fevers in humans. In hyperendemic regions, in areas such as in Africa, there's a high incidence in children under 1 year and also significant perinatal mortality rates. So any strategy to control these diseases must include a control for these Ornithodorus vectors. In this context, vaccines have emerged as a cost-effective and safe method. And in the last few years, our lab has been working on different strategies to develop an anti-Ornithodorus vaccine. Today, I will focus on vaccines based on recombinant protein antigens, mRNA LNP vaccines, and anti-microbiota vaccine. I will start with the first one, with recombinant protein antigens. So what we have done with this strategy is to focus on two key organs in the tick host interface, the salivary gland and the midgut. Why these two are important? Well, they are accessible for antibodies, and also they express proteins that are essential for tick survival. So we have conducted a vaccinomic strategy based on the transcriptome and proteome of these two issues, known as C-alone and me-alone. And based on this data, we have conducted a selection in silico, taking into account different criteria. Then we selected candidates from the salivary glands and for the midgut. Here you can see some of these candidates, antigens that we have tested over the years. You can see here the antigen from Hepaticus, from the salivary glands, and the midgut, and from Ornithodorus mubata. In this case, we have performed an immunization scheme, as we usually do in our lab, based on 3 immunization doses, and 15 days after the third dose, we allow ticks to feed in rabbits. These are the vaccine efficacy for each one of these antigens administered individually. As we can see here, in general, there's partial protection from these antigens in some cases, but it could be good, but none of them has achieved a full protection. So, based on this result, we have conducted another strategy combining salivary candidates and midgut candidates from each species. So, in a form of a multigenic antigenic vaccine. So we did the same immunization trial, and these are the results of the vaccine efficacy. And we can see here that we call the mix of Ornithodorus erraticus. We can see that this vaccine efficacy increased when we administer the combination of antigens if we compare it with the individual ones. But in the case of Ornithodorus mubata, these values are similar, and in some cases even lower than the antigens administered individually. So these results indicate that the combination of vaccine candidates could boost vaccine efficacy, although this might vary among species. Next, based on this result, we start to explore the mRNA vaccines platform. So just to remember how this works, the mRNA of the vaccine candidates are encapsulated in lipid nanoparticles. They enter to the whole cell, and the antigens are produced with similar conformations and post-transcriptional modifications as this native protein. This kind of modification is usually lost in bacteria expression system. So we want to explore this strategy. This is an ongoing project. We only have conducted a proof of concept trial. Here we selected one salivary antigen from each species and other milkweed candidate the same with from each species. So the mRNA synthesis and encapsulation was conducted by a company by GenScript, and we follow an immunization protocol that was reported previously for this kind of platform in pigs. This is the only story so far. So first we evaluate the immune response, the IgG antibody response, and we can see here that after the first dose, the IgG levels of antibody increase. So this indicates that there was a successful uptake of the mRNA by host cell. They were translated to protein, and there was a successful antigenic presentation via MHC2. Regarding the vaccine efficacy, we can see here these are the results, and the green arrows show us how this vaccine efficacy increased when we compare these antigens produced with this platform, and if you compare them with the antigens produced as recombinant protein. For example, in the case of OM86, the vaccine efficacy increased nearly to 40%. So this shows that the mRNA LNP is feasible to develop antiparasitic vaccine. Finally, I will show also last strategy, the antimicrobiota vaccine. This has been done in collaboration with the microbiota group led by Alejandro Cabezas. And we know that the tick microbiome has an important role in the tick physiology and vector competence. And so we have hypothesized that if we could alter a keystone microbial taxa, we could also modify the microbial composition and community assembly. So to test this hypothesis, we have conducted a combination of in silico and in vivo analysis in Ornithodoros mubata. We previously identified Pseudomonas as a keystone taxon that was shared between the salivary glands and the midgut of this tick species. We identified this taxon based on the criteria established by Mateos Hernandez previously to identify keystone taxa in ticks. Next, we conducted an immunization experiment in rabbits. In this case, we used Pseudomonas as a keystone taxa and Lactobacillus as a no keystone taxon, but an abundant one. Also, we include a control group that was administered PBS. First, we evaluate the antibody response. We can see here that after the second dose, the antibody levels, the IgG levels, increase in both groups. However, these were only significant in the Pseudomonas group. Also, we tested the effect of vaccination on Ornithodoros mubata. We see here that only in the Pseudomonas group there was a significant reduction in females, in the survival of females, and in the Lactobacillus group there was a reduction in the oviposition and fertility. Regarding the vaccine efficacy, we obtained that for the Pseudomonas there was a 7.9%, And surprisingly, for lactobacillus, there was a 16.7% of vaccine efficacy. Next, we also tested how the vaccination affected the microbial diversity in Omicron mutata method. We can see here that alpha diversity shows that lactobacillus had the less diverse bacterial community. And regarding the beta diversity, there was a significant difference in bacterial community composition between the control group and the lactobacillus group. So here we can see that vaccination with lactobacillus has a more pronounced effect on the diversity and composition of the microbiome. Then we built co-occurrence network to see how these taxa were interacting between them. So in the Pseudomonas group, we see that if we compare it with the control group, there were fewer interactions and also some changes in the topological characteristics. In addition, we also tested the network tolerance to node removal, and we can see here that the robustness of the network in lactobacillus show a more stabilizing effect despite his reduced diversity. So, as a conclusion of what I have been doing these years, vaccinomics pipeline is a viable strategy for identifying antigen for Remitovirus ticks vaccine. And in this context, multi-antigenic vaccine can enhance protective efficacy compared to individual antigens. The mRNA platform allows experimental validation of theoretical vaccine candidates and can be useful for the development of multi-component vaccines. And microbiome targeting vaccine influence tick fitness and microbial stability. The choice of vaccine target can significantly influence the microbial community within the tick network, and this highlights the importance of studying and understanding the microbiome dynamics in ticks. And finally, the continued research is needed to optimize these strategies and develop more effective vaccines. I would like to thank my research group and also the collaborators. Thank you.
Thank you, Dr. Ana Laura, for your presentation. I see that there is one question for you, but we can answer to that question later in the Q&A session. Okay. So now I will invite Mr. Julio Enrique for his presentation. I believe he is together with Dr. Alina in the room, so I invite them to share their screen.
As I say, our focus is on 4 main applications. The first one is the developing of AI agents for optimization of recombinant protein production in bioreactors. Second one is an app for real-time tick species identification and classification using computer vision. The third application is a multi-platform application for the automation of data management in the National Vaccination Program. And the last one is a chatbot with knowledge base for to support the promotion and digital marketing in the program. So let's dive in. So imagine a bioreactor that learns. We start from the assumption that this is established science. So there are many papers that are talking about it. With some examples. So the objective of this project is that the parameters that are measured by the sensor of the bioreactor can be optimized by an AI. So we are developing and training an AI to autonomously adjust parameters like pH, oxygen, nutrients, etc., to maximize the yield of recombinant K-bag protein production. early trials represent this. You can see at the bottom of the screen the optimized parameter using tested data that you can see in the so this project can arise some technical and operational advantage, like reduce failure and discard batches, like standardization and reproducibility, increase the recombinant protein production via real-time adjustment parameters, and others. So the second app we are focusing is in an app that can identify and classify tick species using computer vision. That responds to the needs before you apply any kind of treatment or vaccine. You need to identify the type of vector do you have. So also we start from the fact that is established science. There are also many papers that are talking about it, and computer vision has established itself as a fundamental tool for pattern recognition in image and videos with a wide variety of applications spanning multiple sectors and contexts. This is a similar application, but it's not available for Cuba. That currently doing the same that we try to do. And this app was trained using images collected from ticks detected in Canada. So we are how does it work? We are training a computer vision model with thousands of images of tick species obtained from international taxonomy databases such as eNaturalis, which will be able to classify species such as Cephalophilus with high accuracy. So this is the model. The objective is to train this model with thousands of images of each species, and possible use of case is a farmer or a user of the app snap a picture or a video. The app autonomously can classify the tick species and provide different treatment and information about the tick. Now, the third part responds to the needs of the national vaccination program to improve the data management. the current data management in the National Vaccination Program faces significant challenges. That data silos are the enemy of progress. The National Vaccination Program faces challenges in efficiently managing the vast amounts of data generated from production to application. Current data collection and reporting models are inadequate, failing to leverage information and communication technology ICTs. This leads to data integrity and availability issues, hindering the generation of reliable reports that inform decision making. So that we are building a live data platform for the GABA program using a cross-platform application with the REST API. On the other hand, can provide more robust and scalable solution for the data management. It can ensure data consistency, provide multiple user collaboration, and offer better performance and security features. These are some screenshots of the mobile app that will be used by the farmer or livestock to provide information about their personal data in the program. And managers and decision makers from the web app can visualize in real time all the data in automatic reports that will have an idea of how the program is doing. These are the impact expected on the vaccination program with the cross-platform application. Now, KVAC is not sterilizing vaccine. It helps to reduce the number of infection by decreasing agarosites. That is shown a digital marketing program. Responding to these needs, we are proposing the development of a chatbot to training with specific knowledge based about the vaccine, the vaccination schedule, to be available 24 hours a day, 7 days in a week, able to respond queries and integrate with the current promotional campaign in the national program. Well, that's all of my presentation.
Thank you, Dr. Julio. Thank you for your presentation. If somebody has questions for Dr. Julio, you can leave them in Q&A box, and we will later reply to them. So now I will invite Dr. Alina Rodriguez Melon for her presentation, and this would be the last presentation of today's meeting.
Okay, thank you, MDAPA, to let us to this webinar. And I want to speak about what to keep in mind when applying anti-tick vaccines in the field. This is the important topic because ticks are more than 900 different species described, and also they parasitize every terrestrial vertebrates, and they cause anemia, toxic effect on the host, and there are direct effects on the host, growth appetite, growth delay, and in general, a diminution in the productive yield in the livestock industry. But the most important concern about ticks is they transmit a wide variety of infectious agents causing diseases, also in humans and also in animals. Here now is that the climate change has changed ticks and tick-borne distribution in the world. And for this reason, is more people is concerned about the problem of ticks. And the principal method to control ticks in the world is the use of acaricides, but they have some limitations because on the animal there are only 5% of the tick population. And for this reason, when you do acaricide treatment on animals, you are trying only a 5% of the population. And for this reason, you do no effects on the population that are on the pasture. And also, this continuous chemical treatment reduce contamination of foods and also environmental pollution, and also the generation of chemical resistant ticks. After 1 year that you are using the same acaricides frequently, you have select a population that are resistant to this chemical, and you need to change this chemical in order to obtain efficacy. And for this reason, the speed in which we develop new chemicals and the speed in which ticks develop resistance is more, and we have the risk to not have chemicals that will be efficacy against ticks. For this reason, we need to use other methods, and in this case, immunological method, we can is a friendly method, and we are thinking in traditional vaccines in which you immunize the animal and you don't have the disease or the infection of the agent. But in this case, we are working with ectoparasites. We have a very big challenge in order to develop this vaccine. In first place, we are a competition with acaricide efficacy, in which when you have a chemical that is has a high efficacy, you eliminate more than 98% of ticks on the animal in the moment after you do the acaricide treatment. But in the case of vaccine, it's not this situation. And the other challenge for vaccines development is the identification of effective candidate antigens. And Laura was talking about this topic, the difficult to identify which will be the effective antigen against ticks. And after that, which you are identifying this antigen, you need to obtain this optimization of the immune response of the host, and also development a method to obtain this antigen in a economical feasibility economical perspective to produce the vaccine. And also you need to go through clinical essay before sanitary register, that we have a very expensive way to arrive to the salient to obtain the commercial vaccine available for vaccination. And the most, in my opinion, the most important thing in this topic is the change of mentality about people that are applying the vaccine, about what to expect from this vaccine. This aspect, we are taking account that the ticks are multicellular organisms and therefore are complex from biological and biochemistry perspective. And they are a very complex life cycles and infest every stage of this life cycle, infest host. And inside of the tick, there are many different proteins that are redundant or complementary functions. And also, there are many proteins that have, until now, unknown functions. And the other thing, important things, is that you are applying a vaccine in order to have an antibody response that able to affect the ticks, but ticks are in contact with the immune system only during feeding. And they have millions of years developing abilities to evade the host immune system. And for this reason, taking into account all these characteristics of things, we are, our opinion is that a single method is not enough effective to achieve the control of the infestation. You need to combine all methods, chemical methods, physical methods, biological methods, and also the immunological methods. The principal things is that together, this the immunological control of the use of any vaccine, you incorporate in this integral strategy to manage ticks. You can incorporate all autonomous practice that there are in any places. And for this reason, you use the vaccine, in this case, the immunological way as a backbone of the management strategy, but the use of chemicals can be not eliminated at all, because one method is not enough effective for this. But in the case that you are using a vaccine, you apply acaricide, but according to infestation, not using chemicals by calendar like as many people use in the world. You do the acaricide bath only 21 days, one a month. In this case, when you apply the vaccine, you use the chemical treatment only when there are more than 10 adult females, engorged adult females on the animals. And for this reason, you expect a diminution in the frequency in which you need to do this acaricide treatment. And for this reason, in this program, when you do application of anti-tick vaccines, you need to do a tick sample in order to identify this species in the place that you apply this program, and also identify the resistant ticks in this area. If you have one chemical in which are not effective against ticks, you discard this chemical and use only chemicals which demonstrate efficacy against this strain, the tick strain in this place. And also, it's important to follow the vaccination schedule in order to maintain the high level of antibody in the animal, in the vaccinated animals. And it's important also do not introduce unvaccinated animals on the farm in order to avoid the recovery of ticks feed on vaccinated animals, recovering his reproductive potential. For this reason, it's necessary to have all animals in the farm vaccinated. And the principal problem that we are having in the field is that the producer, the farmer, are thinking about the chemical treatment in which you do a parasite treatment and you saw it disappear on the animal when the chemical is effective. And also they are expecting the concept of classical vaccine in which you have a vaccination, you don't see any ticks. And this is not the case in this in the fight against ticks, because all the things that I was explaining before. And for this reason, it is important to training people in the what to expect with anti-tick vaccine application in the field condition. In first place, when you have resistant trained ticks in the field, you can use this method and vaccine maybe is applied to all genotype of resistant tick strain, and you must or should expect to reduce tick population. After 2 or 3 generations feed on vaccinated animals, you produce a reduction in the tick population, not only in the ticks of the animal, because with vaccine, the effect is reduction in the eggs line and also in the hatch of these eggs. And after you reduce tick population, in consequence, you reduce the use of chemical in the field. And for this reason, you delay or eliminate tick resistance and extend the useful life of chemicals. And in line with this diminution in the use of chemicals, you decrease environmental pollution and also food contamination. And as overall effect, you obtain a decrease in the tick control cost. Then, in our experience using the vaccination in the field condition, the best results are being obtained when the implementation of this program, including vaccination, is respond to regional policies and also with specialized technical support for training people in the principle of these strategies, and also in the what expect from this kind of vaccination. And in our opinion, the main objective of this program should be tick infestation control rather than tick eradication. Despite there are examples of tick eradication based on the use of chemicals, we think that it's better to control the infestation. And finally, I was to comment some idea on the vaccine characteristics. For example, when you go to select the antigens, develop the vaccine, you need ideally that was effective in all stages of the tick's life cycle and against many tick species. As I told you, there are more than 900 species of ticks described in the literature, and for this reason, you need to select antigens that was present in all the stages of the tick's life cycle, and also effective against many tick species. This is an ideal vaccine, a one-for antigen, and should be easy, relatively easy and expensive to produce the antigen, because taking into account that this is a veterinary vaccine, you need a reduced cost in order to produce the vaccine. And as an idea also is that this antigen or vaccine should decrease the vector capacity of ticks, because this is the main concern about tick, that the transmission of pathogens, and for this reason, decrease the vector capacity of ticks is a very important point when you select the antigens. And also, you need to develop a method in order to obtain a vaccine formulation in order to induce long-lasting immunity, in order to diminish the quantity of booster you need to do on the animals in order to maintain the high level of antibody resistance. These are my commentaries about that, and thanks for your attention. It's able to answer any question.
Thank you, Dr. Alina, for your presentation. So this was the last presentation of this webinar. And now we can answer to the questions that are left in the Q&A box. And there are two questions for Dr. Ana Laura. So first I will invite her to answer the questions.
Yeah, thank you. So I have a first question from Sally Kutler. She said, what was the justification for the use of a rabbit as the immunization model? Curious as Mubata, probably also erratic, will not often feed on rabbits. Well, the target host of the vaccine that we are developing is are pigs. So in the first studies that our group did decades ago, we did immunization experiments in pigs and in rabbits, and we were capable to see immune response against these tick antigens, and also we could see how this immune response influence in the tick physiology. So also pigs, we cannot maintain pigs under lab conditions. It's very expensive. So we established a rabbit as a model for trying our vaccine. This rabbit has low cost and it's easy to handle under lab conditions. And it has worked very well on the Taurus movata and on the Rosa erraticus so far. And the other question is from Alex Rodriguez. Thank you, Ana. My question is, looking at the efficacy result for the antimicrobiota experiment and considering the life cycle of these tick species, what would be the efficacy percentage to say that is useful to use? I think that I cannot say a value for vaccine efficacy, because there's difference between an experiment that you perform on the lab condition and in the field to see what would be the best vaccine efficacy, or at least the number that we could consider a good vaccine efficacy. We will have to go to the field and try these vaccines on the field condition. So what we are doing with antimicrobiota vaccine is that we are exploring alternative strategies to develop a vaccine. As Alejandro was saying, it's just this was only one experiment that we did with Ornithodorus movata. We have to optimize this this strategy. We cannot right now. We are not at the same level that when we combine antigens or even mRNAs. So I cannot say a number of vaccine efficacy. I don't know if that answers your question.
Okay, thank you, Ana Laura. I see there is also a question for Dr. Marcos Andre, so I will invite him to check the Q&A box and answer to the question.
Where can I see the question? Sorry, I'm not familiar.
There is a Q&A box.
Participant anonymous. Okay,
yeah.
I have a question. Is the phylogeny a good indication for a new protozoa species groups, or are there other features that must be taken in account? Thank you. So, that's a very good question. So, regarding paraplasmids, they are quite similar to each other. So we can measure the merozoites and the trophozoites, but this is quite challenging to define species based on morphological features. So at this point, we encourage authors to try to use the near complete 18S RNA to perform the phylogenetic trees in association with several other molecular markers, such as COX-1, 3, HSP70, and the intergenic regions. In the future, I truly believe that we are gonna get more confidence to define species based on whole genome sequence, okay? But at this stage, it's quite hard even get the mitochondrial genomes of these paraplasmids. So I would suggest to use morphological data, several phylogenetic trees based on distinct molecular markers, and if possible, the mitochondrial genome. Okay?
Okay, thank you very much for
Thank you very much
provided
for
answer.
your question.
I have one information for all attendees. I provided multiple links in the chat of this webinar, so you can check all of them. And also I invite you to check the topic that is open for submissions that is related to the title of this webinar. So please go ahead and check a website page of the topic.
So
I think, yes.
Sorry, there's time for one more question or not?
I don't think so. There is no question. Yeah.
Okay.
Dr. Alejandro has to leave, so I will invite Dr. Alina for closing word, and then we can close the webinar.
Well, thank you, Alexander. First of all, I want to thank MDFPA because first by the support for our Congress and this special first volume of our special topic, including these journals, 5 journals of this editorial. And also, I invite all people to contribute to this second volume that we said recently. This was continuous of this special topic in order to publish a topic related with ticks and tick-borne diseases. And also, I would like to chance, all people participating, all speakers, and also all people registered in this webinar. Thank all of you, and we see you next Congress in TTP in 2027 in Portugal. And nice work, and thank you all people. See you next occasion.
Thank you, Alina. Thank you very much. I really appreciated this such wonderful webinar.
Okay. It's a pleasure to meet again.
Thank you very much to everybody for attending. Thank you for speakers for sharing their presentations. Okay, somebody asked me if we can send the links via email regarding the topic. Yes, we can send, but maybe you can also send direct email to me, and I will reply you. I will type my email to you, to the one of the attendees who asked me. So, and all others, you can now leave the meeting, and thank you once again.